Good afternoon. Well, welcome to the Johnson Medical Scholar Series, lecture series. It's a pleasure and honor to have Dr. Linton joining us today. Dr. Linton is a distinguished professor, endowed professor of medicine and pharmacology from Vanderbilt University. He also directs the Lipid research units and the Atherosclerosis research units laboratory at the Vanderbilt University. During his distinguished career, he has done seminal works on the pathogenesis of atherosclerosis, in particular with macrophage cell death, metabolism, and efferocytosis. He'll be talking to us today about this very topic, entitled Macrophage Apoptosis in Atherosclerosis, a matter of life and death. Let's welcome Dr. Linton.
Okay, can you hear me? Is this working? All right, well, thank you. I really appreciate the invitation, and this is, I think, a very interesting program with the medical student involvement. And, and so in thinking of that, when I picked the papers, I was trying to think, like, what medical students, it was hard to really think of something very relevant for them. But anyway, I want to talk a little bit in it of how I got into doing research as well. So, how did I get interested in cardiovascular disease? Well, it's the main thing that kills us, and it's the main thing that kills people in almost every industrial, nation. And really, when I was a resident, I just noticed that there were a lot of people having heart attacks and getting procedures and very little thought about prevention. And while I was a resident was when statins first got approved. And so suddenly there was something you could do, to prevent to potentially prevent cardiovascular events. And, this data from Framingham had been around for a long time that showed the sort of inverse relationship between HDL and cardiovascular events and the direct relationship between LDL and cardiovascular events. And so, if you have an elevated LDL, you have a tremendous increase in risk, but then having high levels of HDL pretty much will nullify that. This is like the killer lipoprotein metabolism slide, but since I'm not sure where you guys are at or if you've been exposed to this, I just want to point out a few of the players I am going to mention. So LDL, comes from VLDL. VLDL is secreted by the liver. The main protein and structural component is ApoB100. It gets metabolized in the bloodstream, by lipoprotein lipase to form LDL, and the other protein on there, though, is ApoE. And so ApoE is produced by the liver to a large extent. It's also produced by macrophages, but ApoE can mediate receptor-mediated clearance of lipoproteins. So can ApoB. ApoB100 mediates clearance essentially only of LDL, whereas ApoE mediates clearance of the triglyceride-rich lipoproteins. ApoE is also on HDL, and our studies really were among the studies that showed that it's important in mediating cholesterol efflux from macrophages. And then one more thing, I'm going to talk about is SRB1, and SRB1 is a receptor for HDL. That's how it's the receptor-mediated uptake of cholesterol esters from HDL, the complete reverse cholesterol transport. But SRB1 is also expressed on macrophages and probably has a much different role in macrophages that I think is important. So here's some cartoons of atherosclerosis that were drawn by my son, Edward Linton, who is a fourth-year medical student at Penn who's applying to ophthalmology and starting to interview. But he took a year out and did macrophage research, so I didn't feel bad about asking him to do this artwork because he was interested in macrophages. And so he basically did all the illustrations for this online textbook chapter we wrote on atherosclerosis. And so I think it's important to start here. I mean, one of the main, ideas of the initiating events in atherosclerosis is this theory called the response to retention hypothesis, and that is that LDL enters the artery wall, gets stuck, gets modified, and that causes formation of oxidized or modified LDL, adds to endothelial dysfunction, which results in release of cytokines and chemokines that attract monocytes, and you get this rolling of monocytes, and then they transmigrate into the intima of the artery wall, and inside the artery wall, monocytes differentiate into macrophages, and they take up cholesterol. So they can take it up either through receptor-mediated ways or also by pinocytosis and phagocytosis. But when the macrophage becomes overloaded with cholesterol, basically when its ability to get rid of cholesterol is overwhelmed by the uptake, then you form these foam cells. And really that's the first recognizable hallmark of a fatty streak, is the accumulation of macrophage derived foam cells in the lesion. I mean, you do have participation of other cells. You have dendritic cells and T cells and also B cells, but that's basically the formation of the fatty streak. And then in progression, you get recruitment of smooth muscle cells, to some extent in response to cytokines produced by inflammatory cells. You get recruitment of smooth muscle cells, and they come in, and they both can turn into foam cells. So there's a lot of evidence now that smooth muscle cells form foam cells in atherosclerotic lesions, and there's a lot of debate about which cell is more important. I mean, I think obviously macrophages are more important, and I'm sticking to that because I don't work on smooth muscle cells. But the smooth muscle cell people say they're more important, but I don't think they have very much evidence for it. But anyway, they can, then they also form basically the fibrous cap. They lay down the fibrous cap, which is thought to be a key element of plaque stability. And so this is a stable plaque with a thickened fibrous cap up here, and then here you have a small necrotic core, and when it, what kills you with atherosclerosis is plaque rupture. And so we say that vulnerable plaques are plaques that have a thin fibrous cap. They have increased, inflammatory cells, particularly kind of on the margins of the lesions. You get, and this shows, the role here of formation of apoptotic cells, and then the failure to, do phagocytosis of the apoptotic cells, which is efferocytosis, leads to secondary necrosis, which causes a big increase in the necrotic core. So another characteristic of vulnerable plaques is having a large necrotic core. And then you get, plaque rupture and thrombosis formation, and it's the clot that immediately blocks off blood flow in the, say, coronary artery and causes myocardial ischemia and infarction. And this is, looking at a section of an artery where there's been plaque rupture and thrombosis, and, points out that this necrotic core is basically a graveyard of dead macrophages, and that promotes inflammation and coagulation. The release of proteases cause breakdown of the plaque and promotes plaque rupture. And so these are just some references supporting the idea and data from humans that apoptosis and defective efferocytosis play an important role in plaque rupture and lead to events in humans. So I'm going to, talk a fair amount about ApoE and in this. And so I want to go back to when I was a fellow, at UCSF and starting to do research. Like I said, so statins had been just discovered. I mean, the first genes had been cloned like really only a few years before that. Some of them were cloned at UCSF. There was a huge amount of interest in molecular biology. And also the first gene knockout mice were made literally while I was a fellow, And I was at this place, the Gladstone Institute in San Francisco, and the whole focus of the place at that time was on lipoprotein metabolism and atherosclerosis. And the first year I was there, everybody seemed to be in agreement that the mouse would never be a decent model for atherosclerosis. And it's because, first of all, mice are HDL animals. They hardly have any LDL. It's because they don't have CETP or cholesterol ester transferase. And really all of their cholesterol is in good cholesterol. But there have been studies that show that if you feed mice a high-fat diet, that certain inbred strains form fatty streaks, just that initial stage, but that's as far as it goes. So they knew at the time there were some models that were susceptible to atherosclerosis, but really all you could get were these fatty streaks. And then while I was there, two different groups knocked out the ApoE gene in mice. So this is a model, obviously a model of the ApoE gene showing it has two different domains, and one of the domains binds the LDL receptor and also heparin sulfate proteoglycans, and the other domain is more of a lipid binding domain. So ApoE can mediate clearance of lipoproteins both through the LDL receptor. It actually has a much higher affinity for the LDL receptor than ApoB does. But it also mediates clearance through this other pathway that are called remnant receptors, and that involves another receptor called LRP and also these heparin sulfate proteoglycans. One of the reasons we knew there had to be another pathway was in humans that completely lack LDL receptors. They get very high LDLs, up to 1,000, but they don't get hypertriglyceridemia. So you knew that they could clear these triglyceride-rich particles out of their blood through this other pathway. But so, when they knocked out the gene in mice, the mice got cholesterol levels of around 300, and they developed spontaneous, atherosclerotic lesions even on a normal chow diet, and so suddenly there was this mouse model where they could get complex lesions, and the lesions, very much looked histologically similar to human lesions in the development of complicated lesions. I think it really revolutionized the study of atherosclerosis in mice. I mean, the other model people use a lot is LDL receptor deficient mice. But these mice, you can study them even on a normal chow diet. If you put them on a high-fat diet, it accelerates the lesion formation. The one part about mouse models that doesn't really replicate human disease well is they really don't do much in the way of plaque rupture and the myocardial infarction part. There's a little bit of plaque rupture, but it's not a great model for that. But so, when this happened, we were working on developing transgenic mice that overexpressed ApoB and had models that would get cholesterol levels of 300 with diet, but these mice had much more severe atherosclerosis. And so really, literally, that made me just think, is it really just the cholesterol level here that's causing the atherosclerosis, or could it be that the macrophage expression of ApoE is important, and that when you lose that in the artery wall, it makes them more susceptible to atherosclerosis? And so, literally one day I was swimming and I just thought, oh, what if you did a bone marrow transplant in mice, and as a kind of poor man's way to switch in macrophages that make ApoE. And, at the time I didn't even know whether you could do bone marrow transplants in mice, but I told my soon-to-be scientific partner, Sergio Fazio, about the idea, and he laughed and thought there was no way you could do bone marrow transplants in mice. And then I looked it up, and of course they've been doing them for years, and they've worked out a lot of immunology by doing bone marrow transplants in mice. And it turns out mice are very easy to transplant, and they don't require immunosuppression and the things you have to use in humans. This was really kind of what we knew at the time. We knew that macrophages, when monocytes developed into macrophages, that they increased ApoE expression, and we thought maybe it could influence cholesterol efflux, but we didn't really know for sure. And so we did this bone marrow transplant, and this was right when I moved back to Vanderbilt. So I actually had gotten the mice while I was out there and sent them to Vanderbilt, came back, did the bone marrow transplant, and literally within 3 weeks we repeated the cholesterol levels and they were normal. So, one thing was we had no idea whether the ApoE coming from macrophages would be able to get onto the lipoproteins, the general lipoproteins in the circulation. Now it seems obvious, but back then if you asked people, people didn't know. But ApoE, and the people already knew it could exchange, but you just didn't know. But just that small amount, it's like 5% maybe of the ApoE that's in the circulation comes from macrophages, was enough to completely normalize the lipoprotein profile. So this is an FPLC of a cholesterol distribution, and this is an ApoE deficient mouse up here, with LDL and V. This is this big collection of remnant lipoproteins compared to a normal mouse with the circles, where you can see the normal mouse really only has HDL. And then when we did the transplant, the other thing you can see is that in ApoE deficient mice, the HDL is basically almost obliterated. And you do the transplant, and just in 3 weeks, all of this cholesterol gets cleared and the HDL pops back up to normal. So that was an exciting day. And it basically smacked of gene therapy, and we showed that it essentially totally prevented plaque formation in the arteries, and it was really the first example of cell-based gene therapy to the artery wall, and we published that in Science, and then have used that model, basically ever since. And it's become a widely used model in the field. But what it didn't tell us was, it didn't answer my original question, because what happened there was you totally normalized the cholesterol levels, and so you had to say that the protection was probably largely due to normalizing the cholesterol levels. But what I wanted to know is whether or not the loss of macrophage ApoE in the artery wall was having an effect. And so then we just did the opposite experiment, and we transplanted ApoE deficient bone marrow into ApoE deficient mice. And it turned out then you got an increase in atherosclerosis, and without a change in cholesterol levels. So then you had to say, well, this is due to the loss of ApoE, or in the artery wall. So did I say this backwards? You got an increase in atherosclerosis with the ApoE deficient bone marrow, so that's right. I don't know why this is upside down, but it's been like that. It's been like that for a long time, and I never changed it. It was just like... Okay, so this is just kind of a reasonably updated cartoon of macrophage cholesterol homeostasis. I just want to make a few points. So I talked about this somewhat today with a few people. So the LDL receptor is expressed on macrophages, but it gets regulated by cellular cholesterol levels. And so it basically gets downregulated when the cells start become playing foam cells, and to a large extent doesn't influence things very much. But so scavenger receptors, and there's a bunch of different scavenger receptors, they take up cholesterol in modified LDL in an unregulated way. And that's the problem. They just gobble it and gobble it, even though the cell has already had enough to eat, and then they can't store it well. And so, you know, what happens is inside the cell, you get, you know, the cholesterol gets released, it's free cholesterol basically from lysosomes, and then there's this cycle of esterification by ACAT that forms cholesterol esters, which are what is stored in the lipid droplets. In the lipid droplets, it's cholesterol ester. And I mean, I think the reason for this is that basically it's a benign storage form. I mean, there's evidence, and some of the evidence come from our studies where we knocked out ACAT1 in macrophages and showed that even though it reduces foam cell formation, it increases atherosclerosis, and you actually get cholesterol crystals. You know, you can take pictures of cholesterol crystals that are spearing the macrophages because the free cholesterol, you know, forms crystals, and it basically is toxic. And so, but then, so you have this cycle like with neutral cholesterol esterase in ACAT where you can go back and forth between cholesterol esters and free cholesterol. It's free cholesterol that gets effluxed from the cells. And so you have, this shows, you know, ABCA1 mediating cholesterol efflux both to, you know, basically nascent ApoA1, but it also effluxes cholesterol to ApoE. And you know, I think that's a very important process. Macrophages don't make ApoA1, so any ApoA1 that's in there accepting cholesterol has to have come in through the circulation. And when you think about it, the more dense the lesion gets, you know, there may not be good access for the ApoA1, but the cells make ApoE and that can, I think, be a very important physiologic acceptor for cholesterol efflux. And you know, the cholesterol efflux is what, you know, can keep the cells from becoming overloaded and becoming foam cells. So, you know, really in the first, you know, years after we did that study, we put a lot of emphasis on the impact of ApoE on cholesterol homeostasis, but it affects a lot of different pathways. It's anti-inflammatory, it's antioxidant. It plays a huge role in reverse cholesterol transport, you know, getting cholesterol back from the periphery to the liver for clearance. But it also is important in apoptosis. I mean, when you knock out ApoE, cells are more susceptible to cell death and apoptosis. So this is a cartoon by Ira Tabus, who really has been, you know, I think the biggest leader in this field in atherosclerosis and studying apoptosis and efferocytosis. And he, you know, made this cartoon to sort of try and explain the observations that seem to suggest that macrophage cell death had different effects on different lesion stages. And so that if you just think of it in a simplistic way, which is the way I like to think of things, is that in the early foam cell lesion, basically all you have is macrophages with cholesterol in there. And so if you kill them, you're going to reduce the size of the lesion, right? And the other piece of that, though, is you need the phagocytic cells, the Efferocytes, to come in and gobble that up. But so that in early lesions, increased apoptosis could actually reduce lesion area, but then in advanced lesions, the ability to clean up the mess basically gets overwhelmed, and you contribute to the necrotic core, and that promotes inflammation and progression of the lesions and plaque rupture. And so I'm really not going to go into this very much, but you know, one of the mechanisms, one of the main mechanisms for apoptosis in macrophages to leading to apoptosis is increased ER stress that promotes the unfolded protein response and leads to different pathways of apoptosis. And we did a lot of studies on fatty acid binding proteins that clearly play a role in, if you knock them out, you get increased ER stress and there are actually some therapies that are starting to be developed based on that. But let's go on here to one of the things we've studied in the last few years is the role of AKT. So, I mean, AKT is a major survival signaling pathway. And so we were just interested in, so were a lot of other people, in the roles of AKT signaling in atherosclerosis, and particularly in macrophages. And, as you can see, it influences a lot of different pathways that can lead to apoptosis. Some of them, that are prominent and have been shown to be involved in atherosclerosis, particularly this pathway of BAD and BCL-XL. And also, we have some studies that show that mTOR plays an important role. But so another thing that has gotten a lot of attention is macrophage phenotype, and this is a I think most people agree this is sort of an oversimplification of different macrophage phenotypes, but there's been a ton of stuff published on this, whether you like it or not. And so the idea is that M1 cells are basically pro-inflammatory, and that in response to certain stimuli, you get an upregulation of certain, pro-inflammatory cytokines, and then other things get downregulated, like IL-10, whereas in M2, you get sort of the opposite. You get they're anti-inflammatory, and you get an upregulation of ARG1 and IL-10 and YM1. But so there was a paper while we were kind of working on AKT isoforms and atherosclerosis. There was a paper published in PNAS that showed that AKT isoform expression affected macrophage phenotype, and that basically if you knock out AKT1, the cells get skewed towards M1, and if you knock out AKT2, they get skewed towards M2. This is another view of macrophage polarization, showing that there are other types, but I think it's even probably way more complicated than this. It's just there's a lot of interesting work on this, on really defining the types of markers for macrophage polarization, but really. This is sort of the same cartoon I showed before as far as the pathways that can be affected, but so these are some summaries of some of the studies that have been done to look at these isoforms. So really, macrophages express three main AKT isoforms, 1, 2, and 3. They actually, 1 and 2 are more expressed than 3, but 3 plays a role. These studies, there have been studies that show that AKT1 expression by endothelial cells seems to mediate a protective, pro-survival effect, and that loss of AKT1 in ApoE deficient mice led to severe atherosclerosis, and they saw increased apoptosis in that study. So another group published that AKT3 deficiency actually promotes foam cell formation, but didn't affect apoptosis. Then a group developed LDL receptor AKT2 deficient mice that developed diabetes, and they showed that there was formation of complex lesions. So I mean, we felt like, this wasn't entirely clear what was going on, and so we initially looked at transplant studies in mice, in LDL receptor deficient mice, where we transplanted the mice that were deficient either for AKT1, AKT2, or both. And so what we saw was actually a huge effect of knocking out AKT2 on atherosclerosis in LDL receptor deficient mice. And we, it reduced the lesion area in LDL receptor deficient mice. And AKT1 in those mice, what didn't have a significant effect. I mean, it looks like it's increased, but it wasn't significant. And this just shows, I'm not even going to go through these blots, but that indeed the AKT2 macrophages were skewed towards this M2 phenotype, and you could look at circulating monocytes, and they had increased expression of CCR2, and then in vitro they had reduced migration. And so we think this was really the main mechanism that was going on, because we couldn't find any evidence for an impact on apoptosis. And so either in vivo or in vitro, in our hands, just knocking out the single isoform. So down here, this is a tunnel assay to look at cell death. There was an effect of knocking out either individually, but if you knocked out both of them, then you see an increase in atherosclerosis. And this is actually consistent with quite a bit of data. Like, there's data from fibroblasts in mice that show the same thing. And so, I think that what it shows is that AKT is so important that it's redundant. And even though you can see isoform-specific effects in certain, measures of function, that there's overlap. And as far as survival, if you lose one isoform, you can really sort of make up for it with the other one. And we actually have a lot more data on this now, having knocked out, pairs of alleles, and it's very consistent. So it actually, as far as apoptosis goes, we think it's the total amount of active AKT and not, what isoform that it comes from that matters. Okay, here's artwork by my number 4 son, Elliot. So he started drawing cartoons in March and has this website where he posts these things, and he's interested in biology as well. And I thought, wow, that looks like a macrophage, and eating a bacterial cell maybe, but you're going to eat that, and then, it gets eaten by the macrophage. But so this is the start of my efferocytosis part, where they're cleaning up. And so efferocytosis means to carry to the grave. And this is another cartoon from Ira Tabus, who does such nice illustrations of these things. And, again, just highlighting this point that defective efferocytosis causes secondary necrosis, an increase in the necrotic core, and weakening of the fibrous cap, and thrombus formation. But another thing that we were interested in, obviously a long time ago, since this paper was published in 2002, I said this earlier, that a problem with mouse models of atherosclerosis is they don't really die of heart attacks, at least not in a kind of a reasonable time frame. I mean, if you really study it over time, the ApoE deficient mice do die over like a 2-year period, but it'd be tough to study that. And these mice, when you knock out both ApoE and SRB1, this is work from Monty Krieger's lab at MIT, he cloned SRB1. And anyway, when you knock them both out, 100% of them are dead by 8 weeks of age. And he showed in this paper that they have heart attacks. He said that, they thought that was the reason they were dying. And I think that's true. They do die of heart attacks, and they do get pretty aggressive atherosclerosis. They don't seem to get plaque rupture. So I don't know that it's exactly an occlusive model, which is what people say, but it's still not necessarily a real model of plaque rupture, but at least it's a model where they rapidly, develop heart attacks and die. And one thing that's interesting is if you put the mice on Probucol, which is an antioxidant that used to be approved for treatment of hypercholesterolemia in the U.S., it rescues them. And then you can breed them and they'll live indefinitely on Probucol, which is this potent antioxidant. It got taken off the market in the U.S. because it lowers HDL, and people were worried, well, if it lowers HDL, that's got to be bad. But on the other hand, it also, like in patients who have familial hypercholesterolemia and have tendon xanthomas, it makes the tendon xanthomas melt away. So, I mean, it's doing something good for cholesterol homeostasis. And anyway, so really all the studies that we do on these mice, you have to have them on Probucol to do the studies. I mean, sometimes we take them off the Probucol to let things go back to normal, but then you've only got about 8 weeks before they drop dead. I mean, it's... So just back to this is a kind of a cartoon of reverse cholesterol transport. I really just want to make the point that if you knock out both ApoE, so ApoE mediates reverse cholesterol transport through a couple of pathways. One is, cholesterol gets transferred from HDL to VLDL and LDL, and then those can get cleared by the LDL receptor or remnant pathways. Most people think of this as being the main pathway, which is, cholesterol efflux from macrophages or whatever cell to form these mature HDLs that go to the liver, and SRB1 basically downloads cholesterol esters, and then the HDL can recycle. But ApoE can also directly clear these ApoE-enriched HDL particles. But the point is, I mean, if you knock out both SRB1 and ApoE, you basically knock out the pathways for reverse cholesterol transport. So one obvious idea in these mice is, a big problem is you've eliminated most of reverse cholesterol transport. But another thing is both of the genes are expressed by macrophages, and we already had a lot of data showing that ApoE was important for macrophage cholesterol homeostasis, and there have been studies on SRB1 looking at it as a role in HDL, in efflux of cholesterol to HDL. But anyway, what happens is these mice, the double knockout mice, so the mice that lack both SRB1 and ApoE, basically the foam cell peritoneal macrophages are just tremendously loaded with cholesterol, with increases both in free cholesterol and esterified cholesterol just in vivo. You pull peritoneal macrophages out and they're just already foam cells in the peritoneum. And so, we thought that probably that cholesterol homeostasis was deranged in these mice, but we wanted to look at it in a situation where the cholesterol levels would stay matched. I mean, before this, we'd done an experiment that was like the first one I showed, which is we transplanted in wild-type bone marrow, and it basically, protects the mice from dying and, gets them down to looking like they're just SRB1 deficient. But, in order to eliminate the change in cholesterol, here we transplanted these ApoE deficient mice where both donor groups were ApoE deficient, but one was wild type for SRB1 and one was deficient for SRB1. And then we fed them a high-fat diet for 8 weeks and looked at the, foam cell lesions. And I mean, I think, you can see pretty easily here that, here these are just like bags of lipid droplets. I mean, this is like, I've never seen foam cells in any other model that are quite like this compared to the ones that got just that were wild type for SRB1. And then this, we also did these studies to look at lysosomal lipid engorgement and free, accumulation of free cholesterol in lysosomes, and those were both dramatically increased in the double knockout compared to the single knockout cells. And So, based on this, I basically have already said all these things that, lack of ApoE and SRB1 causes a dramatic acceleration of macrophage cholesterol accumulation, that you get severe dyslipidemia in the double knockout mice, as well as an intercellular defect in cholesterol homeostasis, and probably both contribute to the altered foam cell formation. You get free cholesterol accumulation in lysosomes, which shows there's defective trafficking, and that's basically one of the things we noticed in some of these studies was that the mice had huge amounts of necrotic cores, and so that made us suspect that efferocytosis might be defective in these mice. And actually, I was presenting some of this data at the New York Lipid Club, and Ira Tavis came up and was like, "Well, so have you looked at this yet?" I mean, but and it turned out to be right, that there's tremendously defective efferocytosis. But so this is from the paper that you guys read. So fortunately, I don't need to go through this in great detail because you've seen it, and we're about to wind down here. But SRB1 deficiency increases the necrotic area, and I mean here, a lot of these studies, we did them in two different types of mice. So ApoE deficient mice, where you can just look at the addition of knockout of SRB1 on the ApoE deficient background. We really, studied LDL receptor deficient, recipients, so that we could look at all four genotypes in one mouse model. So wild type, SRB1 deficient, ApoE deficient, and double knockout. And I mean, you can see, there's increase in necrotic area in both SRB1 and ApoE deficient, but it's even much greater when you knock them both out. And this study, shows that the SRB1 binds to phosphatidylserine, which in apoptosis, it gets put on the exterior leaflet of the cell membrane, and it acts as an eat me signal, basically. And so there's a dose-dependent response in terms of binding of SRB1 to phosphatidylserine, and this looked at some of the features of kind of plaque stability. I mean, there was decreased collagen content, decreased fibrous cap thickness, and I've already shown there was increased necrotic core area. And this is shows how he was measuring this cap thickness. It's very labor intensive. And then this is data. This was a technique really developed by Ira Tavis to look at evidence for efferocytosis in vivo, where you use different stains. In here it tells you what they are. You have red stains for tunnel stains, green for macrophage, yellow for macrophage-associated apoptotic cells, and then white for free apoptotic cells. And so, what you're really looking at is the ratio of free to macrophage-associated apoptotic cells. So, and the more free ones there are, the less efferocytosis there is. So here, I mean, you can see that in both cases, there's just this striking, increase in the here in apoptosis, but here in basically defective efferocytosis when you knock out both of those genes. So a lot of these pathways, that impact efferocytosis work ultimately through signaling of RAC1 that affects macrophage migration and all kinds of things. And it's just, like cytoskeletal protein, and this looks at the effect of activation of RAC1, and it shows that it can rescue the SRB1 dependent membrane ruffling. associated with RAC1. And here you can, this is like a quantitation of the data over here, and with this activator of RAC1, here this is wild type versus SRB1, and then when you include the activator, it basically eliminates that decrease in membrane ruffling. And it also, this shows that an inhibitor of RAC1 suppresses efferocytosis in wild type but not SRB1 deficient cells. And so this, I didn't really show this signaling data, but I mean in the paper you guys looked at, we show that there's a signaling pathway that's basically SARC mediated through PI3 kinase to activate RAC1, and with a possible interplay of other pathways from other efferocytosis receptors. And this last little cartoon that Edward drew is shows physiologic or successful efferocytosis over here. With there's a lot more going on in efferocytosis than I've described here, but these are some of the main players. I mean, the receptor that Irritavus has really, shown is critically important is called MERTK. I mean, we also in earlier studies showed that LRP1 plays an important role, and now SRB1, and basically you get this SRB1 mediated signaling to RAC1, and that allows you to have successful phagocytosis of the apoptotic cell into a phagolysosome, and it basically promotes anti-inflammatory cytokine production, versus when it's defective, you get secondary necrosis and increased inflammation that promotes plaque instability and plaque rupture. So that is the idea. And, I think SRB1 functions as an important receptor both for recognition and clearance of apoptotic cells. It does it through this SARC PI3 kinase RAC1 signaling pathway, and it leads to defective efferocytosis, leads to increased necrotic cores and unstable plaques, and that it's an interesting target for prevention of atherosclerosis, which there was recently this paper from Stanford, Nick Leeper, he was at the ATVB meeting this year, was in Nashville for their first time, which was fun, having all these people come to Nashville, and he presented this there, and he's working with Irv Weissman, who I think is a just world famous immunologist, cancer biologist, and so they already had done these studies in cancer. So CD47 is an eat me signal that in cancer cells, if it's defective, you're not clearing the cancer cells. And so they decided to look in atherosclerosis and showed that an antibody, a monoclonal antibody to CD47, basically by masking that eat me signal, makes the cells get eaten, and so it promotes efferocytosis, and they show in this paper that, you get improvement in the, lesions, decreased inflammation, decreased necrotic cores, and I think it's very exciting. I don't think it will basically eliminate, everything related to atherosclerosis, but I think, this is an important aspect of lesion formation that probably our other approaches don't work on very well, and it may add a lot. But, You know, one thing interesting to think about is a lot of these signaling pathways that I've talked about that are important for cell survival are also important in cancer and are often like genes that are mutated and cause cancer. And you know, you have to think about this, and there's a whole area now called cardio-oncology that's devoted to looking at the cardiac side effects of cancer drugs. Mostly that looks at heart failure just because of the examples of them causing cardiomegaly and that sort of thing. But I think, you know, thinking about atherosclerosis might also be important. But what I think is cool about this is it's something that would be beneficial both for cancer and atherosclerosis. And really, if you think about it, it means efferocytosis is probably a better pathway to look at than just apoptosis per se, you know, I think in terms of your goals and the different parts of, you know, arteries versus organs with cancer. But so the other thing I wanted to show was this recent paper by Dan Rader and company, you know, that shows the description of a rare variant in the scavenger receptor SRB1 equivalent in humans, that this causes increased HDL, but you know, they concluded in this paper that it also increases the risk of coronary disease. I heard from some anonymous professors here that some of the students were skeptical about this work, wondering whether or not it had relevance in humans. Well, here's some evidence that it has relevance in humans. You know, people have looked for a long time for mutations in the scavenger receptors, and they really have had a hard time ever identifying anybody who's truly null for the scavenger for SRB1, or in humans it's called SCARB1, but I think it's much easier to say SRB1, so I'm going to keep doing that. So, you know, and to me that means it's very important, and that most homozygotes don't survive, because it's not that people haven't looked. And so Dan Rader was looking at people who had very high HDLs, because you figure that if you don't have SRB1 working on the liver to take cholesterol esters from HDL, that your HDL goes up and you get very high HDL. And he found a few years ago this woman, a proband, who was SRB1 deficient, and he you know, then showed that her relatives, you know, quite a few of them were heterozygous for this mutation, but it's a very rare variant. And so they then used the power of these genomics or GWAS consortia that have been developed to look for evidence for, you know, people who were mainly heterozygous, but, you know, looking for both in these populations. And so they use these large consortia both for that have been used to look at GWAS studies, plus data from outcomes trials, like a lot of the statin outcomes trials. And they through the miracle of meta-analysis, put this together and looked at the effect on events. And, you know, so these people, they have increased HDL levels, but they feel like there's a significant increase in events of cardiovascular events. And so what they say up here is that, you know, the risk, the odds ratio is 1.79, so almost an 80% increase in risk if you have this mutation. These mutations are very rare. If you look at this, they looked at hundreds of thousands of people to find, you know, 34 cases. I'm not, you know, this is way beyond my skill set as far as biostatistics. Obviously, they have really good people working with them. I have a hard time I mean, I think it's great. I think it actually really supports the idea of what we show, that there's an HDL independent effect of SRB1 that you know, impacts atherosclerosis and cardiovascular events. But you know, I don't know how you pick the controls with such small numbers. I mean, they got to have a way of doing it that's rational. But you know, but I certainly think it's very interesting and you know, amazing use of our modern technology. And so with that, these are the people that have done a lot of this work. A lot of the efferocytosis work was Juan Tao. Patricia Yancey is a serious world expert on macrophage cholesterol homeostasis. Sergio Fazio, you know, and I worked together for 20 years, and then he got recruited to Oregon, but he was involved in a lot of these studies. I didn't show Casey Vickers now, his lab is joined to mine and works on microRNAs. I'm not showing any of that data, and Jay Jerome is a real expert on macrophage cellular homeostasis and lipid droplets and lysosomes, and he did all that work. And so, it's almost Halloween, and these are my kids, like back when they used to be kids. And I'm going to end with another one of Elliot's cartoons, which I think is hilarious. But all right.
So it seems there's like two steps in